Hello student, in previous lecture we have studied the anther culture. Today's lecture I am going to talk about the pollen culture. The culture of isolated pollen into a haploid plant in vitro is known as the pollen culture. Introduction about the pollen culture. Pollen culture is a technique of a production of haploid plant by the culture of pollen grain obtained from an anther of a plant under aseptic condition on artificial nutrient media of a known composition. Here development of plantlet from pollen grain is known as pollen androgenesis. The microspore develop into a haploid embryoid or callus tissue that gives rise to the haploid plantlet by the process of embryogenesis or organogenesis. In anther culture, there is a possibility for genetic exchange during the subsequent generation, and plant are not pure line. So, produce and maintain the pure line. Pollen culture is done. Haploid are raised from isolated pollen grain. An entire plant is raised from a single pollen cell. This produces the haploid used in plant breeding program. Then type of a pollen or method of a pollen culture. There are two common methods for a pollen culture. First one is the nurse culture and second one is the float culture. In nurse culture, the pollen grain are nourished by anther, the same species. This method first described or developed by Sharp et al. In 1972. Second one is the float culture. Instead of isolated pollen grain, entire anther are suspended in a liquid medium to promote the pollen growth. The pollen grain grows into a clump of cell within the anther wall and discharge into the medium. The cell clump are separated by using the centrifugation technique and culture on a semi-solid medium. The float culture method was described by Sutherland and Robert in 1977. Here the media for the pollen culture. There is no common media for a pollen culture. The composition of medium varies depending on the genotype of species, age of anther and all factor of a anther. Here the MSN6 and niche media are modified suitably to grow the pollen. Here addition of cytokinin promote the pollen culture growth of a solanaceous plant. Embryogenesis process is done. Addition of 2,4-D promote the callus growth of a pollen culture of a cereal. Sucrose and fructose are very effective in pollen culture. Here the composition of niche media, their component involved in the niche media and their quantity in milligram per liter. Then procedure of a pollen culture. First collect the flower bud, then collect anther, pollen grain isolated from a anther and diluted with the nutrient medium to concentration of a 10 pollen grain per 0.5 ml of a medium. The anther are placed on a medium in a dish. A paper disc is placed over the anther or callus. 0.5 ml of a pollen suspension is transferred to the paper disc. The petri dish is covered with the lid and incubation 55 degrees Celsius for 4 weeks. After that, develop the pollen grain clone, and these clones are transferred to the callus induction for callus inducing media. Then this callus is transferred to the rooting and shooting medium to develop the root and shoot to develop or produce the whole plantlet. This is the diagrammatic view pollen culture using nurse culture technique. Here first isolated anther, then isolation of a pollen grain, then incubation on a nutrient media, incubation for 4 weeks to develop the clone, and this clone which is transferred to the callus inducing media to develop the callus, and then this callus transferred to the rooting and shooting medium to develop the root and shoot to produce the haploid plant. Here the microspore isolation and culture technique. This is the protocol for pollen culture. Flower bud collected, then sterilization of this flower bud, sterilize the anther, excised anther, then smear the anther to produce the pollen grain, then filter the medium, then centrifugation to produce the pellet. In this pellet present the microspore are suspended in a fresh medium. Then microspore planting of microspore embryogenesis processes take place and regenerate the plantlet. Applications of a pollen culture. Overcrowding of a pollen grain in an anther is eliminated and isolated pollen grain are equally exposed to the nutrient medium. Unwanted growth of a anther wall and other associated tissue is eliminated. Develop pre-line haploid plants. Study of metabolic pathway in plant because of genetic simplicity. Plants are all monoploid, so they used to screen recessive mutation in plant. Used to develop the transgenic plant by way of pollen transformation. In Asparagus officinalis pollen culture followed by colchicine treatment gives rise exclusively male plants. Analyze to linkage data, genetic homology and genetic transfer study. Thank you very much.